Welcome to a new episode of Prime Movers, where we sit down with the founders, CEOs, and inventors who are transforming billions of lives. Joining us this month is Dimension Bio CEO Caralynn Collens to discuss the future of tissue therapies, see how 3D printing, material sciences, and digital manufacturing are helping to usher in a new generation of tissue therapeutics. Here's the full conversation. Hey, Caralynn, thank you so much for joining us this month.
Glad to be here. Thanks, Gavin.
Of course. No, thanks for being here. Can you just give listeners a little background on you and what Dimension's working on, just to kind of get them up to speed?
Yeah, so Dimension Bio, we're a regenerative therapeutics company. Basically, that means we're focused on how can we create life-saving treatments for patients by giving treatments that allow for replacing or augmenting function that's lost due to disease or aging or trauma.
Love it. You're kind of an interesting founder. A lot of people who come on the podcast you're kind of an interesting guest because so many of the people we have on the show are founders. It was their original idea, but I'm always fascinated by the people who step into a startup and have to they inherit somewhat many processes and then they have to take it to the next stage. And that's something you did. I was wondering if you can kind of discuss that, how you've kind of stepped into Dimension and just really continue to tell their story and take it to and continue to fundraise and bring, products to market.
Yeah. So it's I was there so early. It feels like a founder. I'm very blessed that the founders call me a co-founder, but it was an unusual story. Their company was there before I was there. It was based on a technology that spun out of Northwestern University, out of the material science group there. And I had the privilege of meeting the academic founders a few years before I joined them as CEO. So Dimension is actually my third startup. I started my first company coming out of medical school more years ago than I'd like to remember. And that was actually a gene therapy company. It was my first dose of how do you build a deep tech life sciences startup. And I've always been around deep tech life sciences startups. That's basically been the theme of my career, either as an entrepreneur or as an investor, as a founder, and even as an investment banker at one point. And when I was leaving my last company, which was actually an advanced manufacturing innovation center, I was looking for an opportunity to blend life sciences and all these exciting things happening at the cutting edge of manufacturing. And that led me to material science, and specifically to biomaterials and tissue engineering. And again, had previously met my co-founders, Ramil Shah and Adam Jacobs, and I dialed them back up and said, what are you guys doing? And found out they had started a company and were looking at how to use the materials that they developed to be able to regenerate tissues, and looked that there was a lot of promise and really just this interesting convergence of biology and engineering. And they, after a few months of dating, I like to think about it, we decided it was time for all of us to partner together and really build what is now Dimension. And I can't believe that was 5 years ago now.
Yeah, it's incredible. We had Michael Hufford from Ligenesis on the podcast a few months ago, and both of your companies are focused on liver treatments, and there just appears to be this increasing demand for liver failure treatments. There's so much interesting research going on. I mean, what do you attribute that to, and just why there's just so much attention to the space right now?
Yeah, it's really unbelievable. I think, liver disease is one of the biggest killers, unfortunately, worldwide. And most people don't even recognize what a big problem it is. I have to say what Ligenesis and Michael are doing are absolutely terrific. And I think Michael is a tremendous founder and CEO. And it's been great to see what they've been able to do for patients. And, likewise, we're tackling liver failure, patients who unfortunately are in an acute crisis because they have lost the function of their liver cells. And the disease incidence is rising, unfortunately, globally. And it's due to a number of different factors. But I'll say one of the things that was most striking to us at Dimension, we are initially targeting patients with an acute crisis due to alcohol-related liver disease. And
Right.
so these are patients who are in acute crisis, maybe because of alcohol or even drug overdose. And sometimes this can happen in very young, otherwise healthy people, which is really striking and surprising to most people. And the incidence of alcohol-related liver disease deaths has been rising. It's doubled since 1999. And now in the last few years, what we've seen is that incidence is rising mostly in young adults and particularly women. There's also a number of different ethnic groups as well, but unfortunately, the COVID pandemic, I think, is one of the biggest factors. And research now has shown, multiple groups have looked at this, that the rising incidence of alcohol consumption, whether it's because of financial crises or because of just different societal norms, is causing this increase in alcohol consumption, which is leading to increased alcohol-related liver disease and unfortunately mortality that comes along with it. So while we see differences across countries, this is a global issue that we're dealing with.
It is, yeah. For listeners who may not be as familiar with cell-based therapies, can you kind of give them like the bio 101 of how exactly Dimension's therapies direct cell behavior and actually regrow tissue in patients?
Yeah. So, cell therapy or tissue therapy, as we like to refer to our platform as, it's a combination of cells and biomaterials to create more like a tissue-like therapeutic. It's really designed to be a curative or life-saving treatment, and it's because you're talking about replacing or augmenting or the cause, the source of the problem. And so rather than targeting the symptoms, you're targeting the source, the cells that are damaged. And
Right.
so in liver disease, your liver cells, your hepatocytes are damaged. And liver function is important in a number of different ways. It synthesizes things, liver makes things, it metabolizes things like other drugs or other treatments, for
Right.
example, and it also detoxifies. And when your liver cells are damaged, you lose all three of those functions. And so cell therapy or tissue therapy provides a holistic answer to that. It's not just going after one of those things like, replacing albumin, which is a
Right.
protein, for example, that the liver produces, but it's actually doing that and so many other things. And we have found, and others have found, that cells or replacing those cells is what allows us to be able to really treat the patient. Now, you also asked about what are some of the cons. So the pros are this is life-saving curative treatments. The cons are we're still in the early days. So we are still dealing with high costs of manufacturing and limited access for patients. We're trying to navigate the regulatory environment, and so that means that we've got work ahead of us. But I think there's lots of reason to be optimistic. We've never seen these types of curative treatments until cell and gene therapy or these regenerative therapies. And, we're the industry as a whole is about 20 years behind monoclonal antibodies. And monoclonal antibodies today is a $250 billion market. Diseases that we thought weren't otherwise treatable, like rheumatoid arthritis or even certain types of cancer, that now we
Uh,
have drugs like
yeah.
Humira and Keytruda to treat. So 20 years ago, they were dealing with the same problems we are today: the cost of manufacturing, limited access, and how do you navigate a new modality? And here we are now. So I can't wait to see where we're at for cell and tissue therapy 20 years from now.
I mean, do you have any predictions for in 20 years from now? What would you expect to see or hope to see?
Well,
Put you.
I mean, that's exactly what I hope to see. I hope to see that
Yeah.
cell therapy and tissue therapies are just commonplace, that it's not about just treating the symptoms, but it's really about providing these life-saving treatments to patients and being able to offer that to all patients, not because they just happen to live in a particular geography but these become just, another modality. We've got small molecules, we've got
Right.
antibodies and other biologics, and we've got cell and gene. And it's
Yeah.
just, part of the menu.
Yeah. Makes complete sense. I mean, you guys, we first looked to invest in Dimension, I really had to wrap my head around all the other advances that really made you guys possible, like 3D printing, material sciences, digital manufacturing. Like, how it had advances in all of these other areas really helped you guys or emboldened you guys? Because I had to really, to understand you guys, had to understand advances in each of those spaces.
Yeah. Well, and that was exactly what drew me to
Yeah.
Dimension, why we're so excited about building it at this time. Yeah, I was thinking about, like, well, why now? Why
Yeah.
now for this company, for this product? And it really is about that convergence I was talking about earlier. And so, 10, 15 years ago, it was not common to have a 3D printer in your lab if you were in biology or even tissue engineering.
Yeah.
That is much more common now. But that's what Emil Shaw, my co-founder, saw in saying, I want to differentiate. I want to do things others aren't been able to do, and got her first bioplotter in 2010, the first one at Northwestern. And the industry overall is still making the transition from 2D to 3D. This idea of doing things in a three-dimensional environment, even though that's how we naturally are, like our bodies are three-dimensional, our cells are three-dimensional, but we didn't have the tools before. And so fast forward, advances in 3D printing, advances in digital manufacturing. So how do we design things? How do we model new designs? How do we use digital manufacturing to aid in quality so that when we're, trying to do something, trying to make something, we're doing it right the first time and every time, using digital manufacturing to do that. But for us, the biggest advancement was the materials. We wouldn't be a company if it wasn't for the advancements in material science. And so we've got a patent portfolio over 40 patents, and those are all on the materials and how we make those and what those are made of and the composition. And materials have been what's allowed us to overcome some of the challenges in regenerating tissues and replacing function of tissues like the liver. And so I'm sure we'll get into a little bit more about that, but that's the biggest advancement for us is then how do we use 3D printing and digital manufacturing to enable these materials to provide the right environment for cells. And that's what allows us to create these treatments.
So incredibly cool. Just amazing. In May, you guys had or released preclinical data, I believe, where you had a poster presentation that really validates your cell delivery system. And it's really a major milestone that for the field. I mean, can you talk to listeners a little bit about it and what data you guys were able to share finally in May?
Yeah, it was a big celebration for us as we presented our data for the first time publicly, and we did that at the ASGCT, the American Society of Gene and Cell Therapy conference. And there were a few things that we showed, but it kind of goes back to, well, what are the requirements of what we're building? So think about like what are the requirements of the product, and when we're talking about building a tissue or a tissue-like therapeutic, there's a few things we're trying to do. One is we need a lot of cells to create a tissue that has enough function. Right. Two, we need those tissues to function when we implant them into the body. And three, we need them to function long term and to be a durable treatment. And that was what we presented, the ability to do all of those things. And I think this is what's really interesting and tricky also about working in liver disease and working at liver failure. I think more and more people are understanding the idea of replacing function with cells in the case of type 1 diabetes. And so a number of companies and Vertex just present some really great data on this and how you can replace islet cells or the pancreatic cells that produce insulin, how you can use cell therapy to replace that function and to treat patients without having to give them insulin, many times a day. And but when you try to translate that, the idea of replacing function for liver disease, there's a few challenges that no one's been able to overcome before. And one of those challenges is the volume of cells that we need to implant. So if you want to replace liver function, which is what we're talking about doing, patients in acute crisis, their liver cells are damaged, that damaged function needs to replace the cell function. A functional cell can replace those functions. You need billions of cells. And when we talk to clinicians, they know this and they say, we're excited about liver cell transplant, but how are you going to overcome the need to get that many cells into the body and get them to integrate with the vasculature and actually be able to get functionality over the long term? And that's where our materials come in. So we were able to show in the data we presented that we could load sufficient numbers of cells onto our biomaterial scaffold. We call this our BioNitum system. BioNitum is like a nest for cells. And so we're able to put the cells and densely pack them like they would be packed in a tissue, densely pack them into this three-dimensional environment. And then when implanting that in the body, we implant subcutaneously, meaning that we implant it just under the skin. So in like a tissue pocket, we don't put it onto the liver itself. So we're putting it in a place outside of the liver, but we've got to be able to direct that functionality. And so it needs to be able to function when we implant it. But then it also needs to integrate with the tissue. It needs to be able to function over time, over weeks to months, to be able to bring a patient out of this acute crisis. And so those three things, the ability to pack the billions of cells that are necessary, the ability to allow for acute viability, and then long-term function. And then ultimately, all of those things allow you to do is to improve survival. for patients. And so we showed those things, and we were able to show that in an aggressive preclinical model where we give an animal a lethal dose of a toxin to basically mimic liver injury that you would see in a patient that would be coming in with severe alcohol associated hepatitis, to be able to show that when we provide our therapy, we can rescue that liver. And we saw a greater than 70% improvement in survival in the animals that were treated with our therapy. So you can see I
Yeah,
get
super
very
exciting.
excited about what we got here. And we've done a lot more since that we haven't been able to announce yet, but stay
Yeah.
tuned because it's
Can't
coming.
wait to hear. Yeah, excited to hear. I mean, can you talk a little bit about the placement as well? So not placing directly in the liver, and like why is that, and why you're kind of placing it under the skin, like the fattier tissue there. Just
Yeah.
kind of the all the discoveries kind of to get you to that point. Super curious of why.
Yeah. Well, so I know one thing, just feasibility for the patient. So these
Yeah,
patients are in acute crisis. They are very sick. And we talked to clinicians. And I remember some of our early calls when we didn't specify that we were subcutaneously implanting it. They'd be like, you can't do that. The idea of the invasiveness that would be associated with
right,
taking this and placing it on the liver. I said, oh, no, we're not trying to do that. We're trying to basically replace the function of the liver for a period of time to allow the patient's native liver to recover. And so we want to place this ectopically in a place outside of the liver. In our case, we're talking about subcutaneous. This is a minimally invasive procedure that a surgeon can do. It's about the size of like a certain pacemakers or other devices that are even used in liver and disease patients, to be able to give them that supplemental liver function for a period of time. And so by placing it subcutaneously, though, we also you need to function. And right away, it's not connected to the vasculature. So you need to allow that to happen. And that's what we've been able to show from a mechanistic perspective, mechanism of action. That's really what the Bionitem system is designed to do, to allow for that initial ability for nutrients and oxygen to reach these cells and for these cells to be functional, but then to very quickly create new blood vessels to be able to provide that new vasculature that's necessary to connect and integrate with the host, and to be able to do that over time. And that was another one of the big challenges. If you've got to put in that many cells, billions of cells, how can you do that in a way that you can integrate with the tissue, integrate with
right.
the vasculature, but not elicit a foreign body response? So when you put something in the body, if we're subcutaneously implanting these tissue-like therapies, the risk is that they're going to be attacked by the body, by the host, because that's what our bodies are meant to do. We attack things that aren't like us. And so we've got to kind of trick the body. And that's what our material system allows us to do, to be able to say, this is not a foreign body, but it's going to look to be like a like substance, and therefore induce a healing response instead of a fibrotic response. It's going to ultimately choke off the cells. And that's something that we've been able to show, that we believe sets us apart from our peers.
Yeah, got it. I mean, what are you seeing on the regulatory front? I mean, there was a recent FDA cell and gene therapy roundtable for asking for like more responsive frameworks, for therapies like dimensions. Have you seen changes at the FDA with the new administration? What kind of are the evolutions you're seeing on the regulatory side?
Yeah. There's a lot happening on the regulatory side. I mean, I think anyone, looking at the public media will see that. I mean, there's a lot of changes happening at FDA from the very top, but throughout the whole organization. And we'll have to see how those play out over time. But one thing that the new administration at the FDA has made very clear is their commitment to accelerating treatments to patients. And how do we get these new curative life-saving treatments like cell and gene therapy to patients? And how can we make sure that the US doesn't lose out on
Yeah,
that ability? Because we are seeing other countries create new, faster ways of being able to give patients access to these treatments. And we say, what the US FDA is saying is, how can we make sure that our patients aren't left behind? And so there was a cell and gene roundtable that was called in June, and where the leadership of the FDA listened to leaders, key opinion leaders throughout the industry, both clinicians as well as entrepreneurs and patients and patient advocates as well, who said that these are the things we need to do. We need to provide better ways of getting these drugs to patients, be able to reduce regulatory barriers, be able to fast track these drugs to patients. And they listened to a number of ideas. And even just a couple of weeks after this first roundtable, there were already changes in some of the regulations with long-term monitoring for patients with CAR T, another type of cell therapy. So I expect there's going to be a lot of changes we're going to see over the next year that's going to be in favor of giving patients access to cell and gene therapies. And so I think we've got a great year ahead of us to see what the FDA is going to be able to do to make this a better, more favorable regulatory environment for us.
Yeah, I think so too. Uh, to wrap up, we can just do the prime movers questionnaire here. You did it before for the blog back in the day. Now we got to get it live recorded here and see if there's been any updates to your answers here. What's the breakthrough science prediction you have for 30 years from now or 40 years from now? We kind of touched on this a bit for where cell therapies are going in 20 years, but maybe there's one outside of therapeutics that you're excited about. Is there any either in your space or another that really catches your attention?
Yeah, well, so actually, I can't go default to my cell and tissue therapy since that
Most
is where
people do. You can, yeah.
well, I'll say maybe a little bit twist on what I said before, and I'll
Okay,
name one other thing. Is that I think, early days of cell and gene and regenerative therapies overall, you see it in oncology, and then oncology moved to autoimmune. But what we haven't seen yet is how it goes to more chronic diseases. And we're starting to see real advances in type 1 diabetes, for example, and Parkinson's disease. But I think we've got a ways to go to be able to address these bigger chronic problems. I mean, problems like liver disease, for example. And so I'll say maybe that's the twist on what I said earlier is how do we go up beyond these more rare diseases and be able to treat more chronic diseases. And I'll add in another piece too, which is in how AI can also help us to be able to do that. And I know that's,
yeah, just
everyone's talking
supercharges
about
it.
obviously about AI, but there really are, when we think about how do we use it to improving our quality systems, how do we use AI to improve our design and computational modeling? I mean, we're really excited as a company about what we can do there.
My personal favorite. Have you read anything lately that's inspired you? Any book recommendations? We can venture into TV too or music, but I'm always fascinated. What's on founders' nightstands? What's the thing kind of getting them thinking at night, or any? Just mostly to fuel my own reading list. I'm super curious.
Well, I love memoirs and autobiographies, and I'm not done with it. But the one I'm reading right now is Nelson Mandela, Long Walk to Freedom,
Freedom.
and I think what's striking me most, and seen movies and such, but like really diving into his story that he wrote himself. One of the things that is striking most to me is how he's so open about mistakes he's made early in his leadership journey, and how small events had just profound implications on his future thinking, and just really getting that in the sense to be inside that brain of a leader. Those are the types of books I love to read and really
Yeah,
get
great.
into.
Yeah, great recommendation. Don't really think of it as like a leadership book, but it absolutely is, and should be. Yeah, required reading for a lot of founders for sure. What's something you look for in a VC? I mean, at PML, we always try to be a bigger value add than just a check and show up in other places. And when you're looking for someone to be on your cap table, I'm sure other founders who are just getting started are super curious of how you think about that, who you want to partner with. I mean, do you have any recommendations for them?
Yeah, I am. I think you got to look for a partner. It's
Yeah,
that's so much more than money. I mean, this is the group or this is the person that you're going to be in the trenches with, and every company is in the trenches at some points. And so,
yeah,
finding VCs, finding investors who can really partner with you, who when they see the high highs and the low lows, they're not going to be startled. They're not going to be afraid about those
yeah,
moments of like, oh boy, we're in trouble. What are we going to do? How are we going to solve this? But instead can partner with you. And I think that's one of the things that attracted me to PML. There were so many people on the team who had been founders themselves, who
understood.
had been entrepreneurs themselves. And so it wasn't, you guys don't freak out when something doesn't go perfectly, but instead say,
We've been
how
through it
can,
before. We've seen it before. Yeah, exactly.
yeah, yeah. So a
Well,
partner, that's what we look for.
yeah. Yeah, we hear that a lot. It makes complete sense. It's not a very common answer on the podcast for obvious reasons. I think that's it. I mean, Carolyn, thank you so much. I really appreciate you taking the time to sit down with us. And I think this has been absolutely fascinating conversations, and listeners really appreciate your insights into the space. And yeah, definitely look up Dimension Bio, no longer Dimension Inc. You guys recently rebranded. Anything on that, or really just maybe talk about that a little bit with folks to close things out, just about the rebrand and why it was necessary to uh, change the name and really focus on or how it defines what you guys are looking forward to doing in the years ahead.
Yeah. So, I mean, for us, it really was just signal where the company is going. But in so many ways, that's how we always started. We always knew that what we wanted to use is figure out how to use our biomaterials platform to be able to treat patients in ways that we couldn't before, to be able to
Right.
provide these new function, this augmented function, this replaced function, and so when we realized that we had that breakthrough and we figured out how we could create these durable treatments and how we could get vascularization and not get the foreign body response that others have been challenged by, that for us was saying, okay, we have a chance to do something and really leverage biology and
Yeah.
leverage biology in a way that people haven't been able to do before. And so that was our stake in the ground. We're Dimension Bio. It's not just about the tool. It's really how does that tool create the therapeutic? And that's
Right.
where we're focused today.
Got it. Well, thank you again so much. Really appreciate your time. Been a
Thank you.
fantastic half hour, and thank you again. Have a great
This has
one.
been great. Thanks so much, Gavin. Take care.
Thanks, everyone. Bye.